Okay, let's look at the roles of auxins in plant responses. So the first one, we're going to look at phototropism, later on geotropism. So a few key concepts here that you need to take note. First is that the response direction of the shoot tip. It actually depends on the direction of the light stimulus. So you have the light stimulus over here. The distribution of auxin in the shoot is uniform if the shoots are exposed to light from all directions, and therefore this will cause the shoot to grow upwards. But when the shoots are exposed to the light from only one direction, contohnya here, sebelah kiri sahaja, so that means it is not exposed to light from all directions. So this region here at the left side gets light more than the right side. Okay, so please bear in your mind, auxin ini tidak suka cahaya, so it will move away from the light. Therefore, there will be more auxin here at the shaded area. Lebih banyak auxin di sini. Okay, remember, auxin punya function, it helps. It serves in cell elongation. More auxin at the shaded side means there will be more elongation as compared to the bright side. That's why the shaded side will elongate more than the bright side. So as a result, the shoot will bend towards the light, and that's why that is how the shoots are showing positive phototropism. Okay, the second scenario is that the coloptile tip is being cut. Okay, what's in the tip? Very important to know, to realize that auxin is produced at the tip. So if you cut the tip, that means you remove, there's no more auxin, you remove the auxin already. Okay, no auxin, that means no cell elongation, therefore no growth is being observed. Okay, the third scenario is that you have the tip being covered with opaque cover. Okay, jadi it seems like the shoot now is having uniformly exposed to light daripada semua arah. Okay, so if it is uniformly exposed to light, okay, what will happen is that it will grow upwards lah, like this first scenario. Okay, like the key concept here. Receiving lights from all direction, therefore auxin dekat sini taburannya adalah sama, and hence it grows upwards. Okay, the fourth scenario is somewhat like the third scenario, just that the cover is now transparent. Okay, transparent means cahaya boleh masuk. So seolah-olah the first set sama dengan control set. So therefore you will get to see the same observation as the first set. Okay, here the fifth scenario is that you have the base of the tip, bahagian tapak, bukan hujung atas, tetapi tip of the base is being covered with opaque object. Okay, this one is observation after 2 to 3 days lah. After 2 to 3 days. So what are you going to see after that? After 3 days, the coloptiles bend towards the light. So seolah-olah sama seperti the control set and also the fourth set, which means tidak ada effect ya. Auxin masih ada di sini. Auxin go diffuse down, and then because it is light fearing, dia lebih banyak dekat bahagian shaded. Okay, this region elongates more than the bright side, and therefore it bends towards the light. Okay, now the coloptile tip is separated by the piece of agar block. Agar block. Okay, so after 2 to 3 days, same observation as the control set. So maksudnya tidak ada effect. Auxin still can diffuse downwards menerusi this agar block to the zone of cell elongation. But the last scenario is that now it is separated by mica. Okay, this piece of mica, and it seems that no growth is being observed, meaning auxin, which is from the tip, is unable to diffuse downwards across this mica. So mica is the metal. Okay, it's the material that is now blocking the diffusion of auxin from the tip to the zone of cell elongation. Therefore, you don't get to see any growth. Okay, this is on phototropism. Okay, the second role of auxin is in geotropism. because auxin also controls plant response to gravity. Okay, and the seeds germinate horizontally in the soil due to the pull of gravity. Sebab ada tarikan gravity, so auxin pulls at the bottom side of the tips and the roots. So dia akan berkumpul di bahagian bawah sini because of the pull of gravity lah, and then the roots are exhibiting positive geotropism. Roots. Meanwhile, the shoots grow upwards against the pull of gravity, exhibiting negative geotropism. Okay, so in the shoots dengan in the roots different. You still remember we have this graph, okay? This graph, growth response. Okay, stimulation and inhibition dalam keadaan di akar dan juga pada shoot. So as the concentration of the auxin increases in the shoot, it serves to stimulate elongation, but then in the roots, it's going to inhibit. Again, this is what happened here in the roots and also in the shoots. So high auxin concentration at the bottom stimulates elongation. There's more auxin over here, right? So this region will elongate more, and therefore the shoot tips bend upwards. But then in the roots, high auxin concentration, refer the graph just now, terlampau banyak auxin ataupun kepekatan auxin tinggi, dia akan merencat, inhibit cell elongation. Okay, cell elongation akan terencat. Okay, and then the roots is going to bend downwards. Okay, maksudnya ini adalah berlawanan dengan shoots. Okay, one stimulate, the other one inhibit cell elongation. Okay. Okay, now we continue to look at other phytohormones. We have gibberellin. Okay, gibberellin is essential in germination. Tengok gibberellin. Okay, germination of the seeds, growth and elongation in the stem cell, developments of leaf, flowers, and fruits, inhibits development of roots, and also causes growth of flowers in dwarf plants. Okay, cytokinin. Look at the key concepts here. Cytokinins promotes mitosis. Okay, stimulates division and also elongation. Okay, with auxin present lah. So stimulates seed germination ini is very essential in cytokinin. Okay, more mitosis. Here you see, cytokinins, more mitosis. More mitosis means there'll be more cells being produced. More cells means more growth. Okay, so there's a word over here that cytokinin delays leaf senescence. Delays tu dia melambatkan proses penuaan. Senescence is proses penuaan, process of aging. Okay, and also it inhibits development of apical dominance. Bahagian apex, bahagian hujung itu dia merencat perkembangan the apical dominance. Okay, so remember cytokinin, remember mitosis, remember that it stimulates cell seed germination. Okay, next one is abscisic acid. Okay, abscisic acid ini is very special because like you see, it induces seed dormancy dekat sini in this table that you can see. Abscisic acid, seed dormancy. Okay, dalam keadaan yang tidak sesuai, dia akan merangsang seed dormancy. Okay, so it will induce stomatal closing during drought. Keadaan kemarau, it inhibits the growth of buds and also seed germination. But it stimulates abscission of mature fruits, leaves, and flowers. Okay, it inhibits growth of the plants. So masing-masing ada dia punya fungsi yang khas yang kamu boleh ingat untuk setiap satu phytohormones ini. Okay, ethylene is very essential in ripening. Okay, so it's going to stimulate ripening in the fruits. It stimulates the senescence process in the plants, and also it stimulates abscission of the leaves and also the fruits. Okay.